Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Unlocking the Transcriptomic Potential of FFPE Cancer Samples, a Cross-Platform Comparison Study, and our sponsor is Lexogen. Our panelist today is Dr. Arran Turnbull, Postdoctoral Research Fellow for the Edinburgh Breast Cancer Now Research Team and Group Leader of the Translational Oncology Research Team at the MRC Institute of Genetics and Molecular Medicine at the University of Edinburgh. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. If you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Turnbull. Please go ahead.
Good afternoon or good morning, depending where you are in the world, and thanks very much for joining me today. I'm going to start with you by introducing myself. I'm a breast cancer researcher. I've been working in breast cancer for about 10 years. I'm going to say up front that I'm not a technical expert in the chemistry of sequencing, nor am I a bioinformatician. Where I'm really approaching this from is as a translational researcher. I have a clinical focus with an interest in bioinformatics. And my research over the last 10 years has involved the use of clinical patient samples and largely transcriptomic analysis. So when we started about 10 years ago, we were using fresh frozen tissue samples. And to be honest, the technologies available at the time had high-quality RNA inputs, and we didn't really have any option for FFPE tissue. But FFPE is an enormously valuable resource. They're collected routinely clinically. They're stored at ambient temperatures for many, many years, and for us, we have a tremendous amount of follow-up. So there's a wealth of available information and data that we can get from FFPE-derived RNA. So as a researcher approaching this question, my main focus was, can I get usable data from FFPE tissues, especially older tissues with maybe 10 or 20 years follow-up when we know that RNA degrades over time. So this is not going to be a technical or a very comprehensive comparison of these technologies. This is basically going to be a series of trial experiments that we performed in order to find a technology that would address our needs. When we discovered how many of these samples we had across different technologies overlapped, we felt the most appropriate thing to do was to analyze these as one data set. And that brings me on to my talk today. So really, I'm going to frame this in two questions. And really, the purposes of this is to share my experiences with a range of these different technologies to study clinical FFPE tissues. So I suppose the first question we have is really the one you see there in the green box. Can we unlock the transcriptomic potential of archival FFPE tissue samples and derive usable transcriptomic data? And when we have that data, if we think about the blue question now, can we integrate that with existing fresh frozen data, with other FFPE RNA-derived data sets for a more of a meta-analysis type approach? So these are the questions that I'm going to be discussing today. I'm going to be talking about a range of technologies. So as I say, when we started down the route of transcriptomics and breast cancer some 10 years ago, we were working with microarray technologies available at the time, the early Affymetrix chips, the early Illumina chips. And when we decided we wanted to move into the wealth of data available from FFPE, we had to go looking for a technology that was going to deliver for us. So we've tested a wide range of technologies, and I'm going to take you through our analysis of those today. So first of all, a little bit of background. We know that since their inception, microarrays have been adopted as a major tool for transcriptomic study of clinical samples. We've been using them for years ourselves. And this has really helped us to understand how disease develop, how we can start to molecularly subtype and derive prognostic signatures from different clinical samples, which can really have an input in translational research. A crucial consideration for many researchers into clinical studies is whether the data that they derive from one study can be combined to another study for a reliable meta-analysis. So I want to start by just explaining this one example to you. This is from a paper back in 2000 and 2001. And this is an example of a data set that I'm going to come back to and use again later in the presentation. So in this data set, what they did is they took some fresh frozen high-quality RNA from breast cancer samples, and they performed microarray analysis at the time and derived a gene expression signature profile which could divide all breast cancers into what we class as the intrinsic subtypes. So you can see the dendrogram in the top left there has the various intrinsic subtypes as they've been defined. And these are clinically relevant subtypes which translate into survival and recurrence-free survival, as you can see the plots at the bottom. So we can basically take any breast cancer data set and apply these intrinsic set of genes to define and classify cancers as belonging to one subtype or another subtype. And this is an approach that I'm going to use again later in the presentation. So the question is, why would we want to integrate? And as I said before, this really allows us to perform a meta-analysis and clearly allows us the possibility to improve our statistical power and our generalizability of results to make more robust classifications and response predictions. One other advantage is we can start to make use of the vast quantity of publicly available microarray and transcriptomic data which is out there in the public domain. One of the issues with integrating data from different studies is what we call non-trivial systematic bias or batch effects. And these occur within experiments or between experiments. And as a group, this is something we've been interested in for many, many years now. So I'm going to take you back to our paper in 2011. And this is really a technical study we performed using universal human reference RNA, so one single control sample. You can see the flow of that sample on the left-hand side here. We extracted RNA, we converted to cRNA, we performed amplification and labeling across a series of experiments. You can see the one experiment in the green box on the left-hand side, and the second experiment on the red box. These are performed different times, different dates. The green experiment was processed on the Illumina Human Ref 8 chips, and the red experiment on the Human HT12 chips. So slightly different platforms as well. And what we basically wanted to ask is which of these different contributing factors from the processing of that initial RNA extracted sample to the point where we're actually putting these things onto chips and begin to analyze the data, do we get a bias, and where does the bias come from? So if we now look at the heat map on the right-hand side here, let's look at side A first of all. What we basically have is the samples. You can see the green box along the bottom there and the red box up the middle. The colors there correspond to the individual chips which were run on the left-hand side in the green and red boxes. So we're basically looking at every sample against every sample. And when you compare on plot A there on the right-hand side, the red box, which is the samples on the second run, with the green box, which is the samples on the first run on the Human Ref 8 chips, then what we see is significantly reduced correlations. So these are Pearson correlations. So despite the fact we are starting with the same sample, had we processed that same sample within the same experiment, or within two different experiments, we are introducing a difference. We're introducing a systematic bias. When we quantile normalize this data, as we've done on the right-hand side, we actually cannot remove this bias. We still see this difference in the data, which is simply due to the way that the experiments were performed. And essentially what we can say is that the inter-experiment bias, so the bias between the individual experiments, is the biggest contributor to the bias that we see in our data, despite the fact that these are identical samples. So what we've done here is taken a series of publicly available data sets. So these are breast cancer data sets. And this is a paper from our lab in 2008. And what we've demonstrated here is that this bias, which can occur because of the way samples are processed in the lab, can be successfully minimized, allowing for the integration and analysis of breast cancer data sets for a meta-analysis kind of approach. So what you can see here, this is multidimensional scaling, and we have a series of data sets here, put the references in there. And these were performed on a range of different Affymetrix platforms. So each platform there, or each data set, sorry, is a different symbol. And what we've done is classify these tumors by that intrinsic set of breast cancer genes that I discussed in the first couple of slides. So it's the same 496 genes that we're using. When we put all these data sets together and cluster them, as in that left uncorrected picture, what you see is that they largely cluster by the data set that they came from. They're clustering there by data sets. What we've done on the right side is simply apply a simple mean-centered correction of that data. After doing so, we now begin to see that you can no longer distinguish the different platforms, or rather the different data sets, the different Affymetrix data sets. What you can now see is the true biology. So that the different colors, of course, denote the different intrinsic breast cancer subtypes that we defined in the earlier slide there. So we're now beginning to see the biology, and this is quite a simple, straightforward change that we can make to the data. And if we look at the next slide, this is a paper that we produced in 2012, and this was more of a technical study as well, following on from that initial work that I just showed you. What we took is fresh, frozen breast cancer tissues from 6 patients. So these are patients that were given a particular treatment. They were given this before their surgery, and we took a biopsy. Of the tumor at diagnosis, 14 days on that treatment, and at 3 months on that treatment from each of these 6 patients. So we had 18 samples in total, and we profiled these on the Illumina HT12 bead arrays and Affymetrix U133A chips available at the time. So unlike the previous examples, when we're largely using different chips from within the same manufacturer, in this example we're using 2 completely different microarray approaches. And we'll talk about the approaches later. So what we demonstrated in this paper, this is a figure from that paper, is that once again we can minimize the bias that is introduced simply by having the data produced from different studies and on fundamentally different technologies. So again, what we're looking at here is pairwise Pearson correlation heat maps. So we have, let's just orient you to this. What we have is we have the pre-treatment. If you look at the left-hand side, the pre-treatment samples, the 10 to 14 day samples, and the 3-month samples. The green represents the Affymetrix processed samples, and the blue represents the Illumina processed samples. Now, if you actually look at this sort of cheese toasty kind of plot here, what we have is the green sample, the green letters or the green samples, they represent the Affymetrix data, and the blue samples represent the Illumina data. Now, these are in order. They're in a numerical order. So patient 1, sample 1, 2, 3, patient 2, sample 1, 2, 3, and so on. Oh, sorry, someone's changing the slides here. Sorry about that. Someone is changing these. Let me go back to where we were.
We're sorry about that, Dr. Turnbull. We had a slight technical issue, but we should be back on track now.
Oh, perfect. So what we actually have here is the arrows are corresponding to the color, and they show the order. So we're going from the same patient up to the final patient through samples 1, 2, and 3. And what I haven't shown here is the raw data. If we did no correction and we had the Affymetrix data and the Illumina data here, it would be completely off the scale. There's a massive, massive difference between these 2 data sets, and I'll be demonstrating that more clearly later on in the presentation. This is after using 4 different correction methods, and really what you need to draw your attention to is the inner yellow square of diamonds. And what these represent are the Pearson correlations between matched samples on Affymetrix and Illumina after the different correction methods. So each quarter of this chart is a different correction method. You can see XPN on the top left, mean centering on the top right, which is the same approach we used previously. You can see distance-weighted discrimination on the bottom right, and then combat correction on the bottom left. Now, what's quite interesting here is that the correlations between the matched samples across platforms with both combat and XPN are very strong here. This is quite a bright yellow. This is close to a Pearson correlation of 1. Whereas the mean centering is not quite as good, and with distance-weighted discrimination, what we've actually done here using this approach is overcorrected the data. So we're no longer seeing that difference between the background data and the matched samples. So The purpose of this paper was really to demonstrate that even with those older technologies, using established correction methods, it is possible to combine data and derive reliable and useful transcriptomic data that can be analyzed. So the correction methods that I mentioned, these are really two correction methods which we found in that paper to perform the best, and we've used these quite extensively to fix this specific problem of systemic bias that we see between gene expression platforms. I'm not going to go into the underlying mathematics of these methods, just to say that we use these methods as described in the original publications. These methods have been used and cited several times. One of the advantages that we found to using Combat is that it's suitable for small data sets or for unbalanced data sets where you have different sample numbers across different data sets, and you can actually process more than 2 data sets within the same iteration of the algorithm. For XPN, you need a certain number of data sets, a certain number of samples in each data set, and they should be quite balanced. So there are available methods for the reduction of systematic bias. This is another example from that same paper. The differences here are between 2 published data sets. Again, one was on Illumina and one was on Affymetrix. I put the references for these studies there. And essentially, we are using the same 496 genes here, as I showed you in the initial slides, to classify these patients into their intrinsic subtypes. And what you can see in panel A at the top there is that really the dendrograms represents the different data sets. The one on the left is the Illumina data set, the one on the right is the Affymetrix data set. All the samples within each of these data sets have been classified to a particular intrinsic subtype, and in this case, the reason there's 3 lines underneath there is that we have used 3 different approaches to classify samples. But you can largely see the red samples, which represent the basal subgroup, are clustered all near each other. Then we have the blue, which are the luminal B, and then the darker blue, which are the luminal A. But each of these data sets is clustering independently. So the biology within the data sets seems to be fine, but they are clustering independently. After correcting these data sets with Combat, in this case we used the Combat method, you can see in figure B here, where now all of the basal intrinsic samples, the red ones have come together, the light blue ones have come together, and the dark blue ones have come together. So we have significantly increased the sample number in this data set available for a meta-analysis. So further details of this work are, of course, available in that paper. And really, I just want to summarize what I've said so far about integration before I move on to these new technologies. So direct data integration allows for a robust meta-analysis, and we have the potential to improve statistical power and, importantly, make use of publicly available data. I've demonstrated some non-trivial systematic bias or batch effects, which can occur from various points within the processing of samples, most importantly between different platforms and between different independent experiments. Correction methods exist which can reduce that bias and allow the true biology to be elucidated. One of the caveats, which I'm not going to talk too much about here, but which we do refer to in the paper, is that the success of integration of data sets completely depends on selecting appropriate data sets for integration. For example, if you're looking at a treatment effect, you don't want all of one treatment in one data set and all of another treatment in a second data set. It's got to be appropriate data sets to be combined to address a particular scientific question. So this brings me on really to the next part of the talk, which is about fresh frozen versus FFPE tissue. And as I said at the beginning, as a researcher, this is my main question. In translational research, I have access to huge numbers of FFPE tissues with a wealth of clinical data, and up until this point, I haven't been able to derive transcriptomic or reliable transcriptomic data from that resource. So as I said before, traditional microarrays required high-quality RNA, and for that reason, for years, we collected fresh frozen tissues. Of course, the problem with that is that there are logistical challenges with the collection of fresh frozen tissue and their long-term storage. What we really need is a reliable technology that can harness the valuable information available in FFPE tissues. So the question is this an option? Do we have, or can we use FFPE tissues? So a large number of studies have compared this very question before, these matched fresh frozen FFPE samples. And some have reported reduced efficacy or numbers of detected transcripts and batch effects, quite similar to how I explained a second ago, between these two different matched samples, between the FFPE and the fresh frozen. Most studies, however, conclude that there is data generated using FFPE can compare to the original fresh frozen to some extent. What's really promising is that newer kits and newer sequence-based technologies have emerged which hold real promise for the processing of these FFPE samples. So new technologies really stemmed, I suppose, from RNA sequencing. And we've done a bit of RNA sequencing ourselves. And really, I think this has probably supplanted microarrays for transcriptomic analysis. However, RNA sequencing is not without its challenges, or full RNA sequencing is not without its challenges, and with associated costs. And as translational researchers, we are often interested in differentially expressed genes and pathways, and really integrating the transcriptome. And fortunately, there are now a range of new and emerging technologies which seek to address this problem. And we essentially wanted to find the most appropriate technology for our purposes, and so set about testing various available technologies and in terms of their ability to produce robust and usable data. So this is the figure I showed you before. And again, as I say, I'm not an expert in the chemistry of sequencing. What I should say is most of these technologies and most of the sequencing actually was performed at the genetics core at the University of Edinburgh, with the exception of Affymetrix and nanostring, which was performed at the host and tumor profiling unit at the University of Edinburgh. So right up at the top here, we have in the yellow box, we have our traditional sort of newer kind of microarrays. We have the old Illumina microarrays, which we were using for years. And then we have the newer Clarium S microarrays with some of their newer kits for processing FFPE samples, which we were initially interested in. We then moved to targeted sequencing, such as AmpliSeq RNA. BioSpider sequencing is an interesting approach which uses direct sequencing of the RNA in an FFPE sample. Of course, we've got full RNA sequencing. And then we have the 3' RNA sequencing technologies, Lexogen, QuantSeq, and QIAGEN. And then we have the gene expression assay NanoString. So the first figure here really documents all of the studies that we have processed on these samples, not just the study or the overlapping samples that I'm going to explain in a second, but really every sample, every FFPE sample that we have processed on these technologies from various studies over the last 2 or 3 years. Most of these are breast cancer studies, and really what we're indicating here is the date or the age of the FFPE tissue on the left-hand side. So we have some newer FFPE tissues that were produced in the last 2 years, and we have some archival tissues that were produced more than 10 years ago. We've got each of the technologies listed there in order, from Illumina, Affymetrix, AmpliSeq, BioSpider, RNA-Seq, Lexogen, and then QIAGEN, QIAseq. The green lines represent samples which passed the manufacturer's recommended QC criteria for sequencing. So these are different in each case, but things like read length, sequencing quality, and read depth, and other metrics such as that. And the red sample, or the red lines, indicate samples which have failed those QC criteria. So you can see right at the top, the old-fashioned Illumina bead arrays perform very poorly with FFPE tissue, very poorly for older tissues, and very poorly even for more recent tissues. With a collaborator, we performed some Affymetrix microarrays with the Sensation Plus TM-FFP amplification kit, and these actually performed well for that series of samples, but are not without reasonable cost. We then looked at AmpliSeq RNA. So this is an approach where the primers really can appear anywhere within the length of the transcript. the pairs of primers, and we had reasonable success, but less so with older samples, so FFPE tissues more than 10 years old. BioSpider was an interesting approach. Again, this is the approach where the sequencing is performed directly onto the RNA. We have only performed a small subset of patients with this, but that worked quite nicely. Again, we've performed RNA sequencing, which is full RNA sequencing, Illumina sequencing, which is, of course, costly, and we've only performed this in a small number of samples. And these were samples that we were interested in actually getting some mutational data from. We've performed a number of studies with Lexogen QuantSeq, including over 180 samples, which are more than 10 years old, and successfully generated reliable and usable transcriptomic data from those, which has been fantastic. Again, QIAseq is another 3' technology, which again we've performed a small number of samples with, about 50, and again the results were promising. So I'm now going to really move on to the meat of this presentation, which is our comparison across these different technologies. And what I'd like to do is just give a tiny bit of background about the samples here to try and to help you interpret the results that we've discovered. So again, as I said, our main focus is in breast cancer here, and around 75 to 85 or 80% of breast cancers are hormone sensitive. So essentially, their growth is driven by the hormone estrogen. And endocrine therapy is a drug or a series of drugs which can block the effects of estrogen and are used routinely in the vast majority of breast cancers as a treatment option. So what we've done here is we have 11 breast cancer patients with the same approach as before. We had a biopsy at diagnosis before treatment. We had a biopsy at 10 to 14 days on treatment, and then we had a biopsy at 3 months on treatment. Now, these are all neoadjuvantly treated patients, which means endocrine therapy given before surgery. And we determined clinical response based on 3D ultrasound image changes or size changes in the tumor based on the ultrasound measurements. And as you can see from the graph on the right, which shows the relative tumor percentage volume going across days on treatment, all of these patients responded well to endocrine therapy. Now, we've done a lot of transcriptomic work before, looking at the effects of endocrine therapy in patients that respond well and patients that respond less well to therapy. So we're familiar with what kind of changes we would expect to see. Indeed, we've published quite extensively in this area. So if we think about the response to endocrine therapy, these 11 patients I'm going to talk about were selected from a much larger group of about 30 patients who all received the same therapy, and they all responded well to endocrine therapy. And what we defined in our study in 2015 is a series of genes, 42 proliferation genes, which are the expression of which reduces very early on endocrine therapy. So we can see 2-week reduction, and the gene expression stays low at 3 months. So if we look at this heat map here, what we have is all 30 patients in chronological order, patient 1 to 30 along the top. We then have the same at 2 weeks, 1 to 30, and then the same at 3 months, 1 to 30. This is just simply an expression heat map of log2 gene expression data, where red is high and green is low. So we can see proliferation is higher in the pre-treatment samples, and then significantly lower at 2 weeks on treatment and 3 months on treatment. And this really corresponds nicely to the clinical response that we see with the ultrasound measurements. So our study of comparisons, what we have is, from these patients, we have matched FFPE tissues and fresh frozen tissues. So from these 11 patients, when the biopsy was taken, half the biopsy was put into liquid nitrogen for fresh frozen, and half was processed as a formalin-fixed paraffin-embedded block. We extracted RNA from both the FFPE and the fresh frozen, and we profiled these on the range of technologies that I discussed before. So a few caveats here. The Affymetrix U133A arrays are not whole genomes, so we're not included in all the analyses. The Affymetrix Clarium S array, we only included 1 patient on that, so again, that's not included in all the analyses. And of course, the nanostring is not a whole genome technology, and so we only profiled a panel of 50 genes. So I'll come back to them at the end. The other caveat here is that this was not designed to be a technical comparison study. So we don't have every sample on every platform. We performed quite an extensive suite of trial experiments in these different technologies, and then later decided that there was enough of an overlap to perform this study and just have a think about integrating these kinds of data. So this is really the table which shows across which technology, which samples were present. So again, we have our 11 patients across the top. Most of these have a -1, a -2, and a -3 sample. That's baseline, 2 weeks, and 3 months. The pink samples are all the FFPE tissue, and the yellow samples are fresh frozen tissue. So again, our fresh frozen data was, our fresh frozen RNA was processed on Illumina. We did full Illumina RNA sequencing with some of those samples, and for our new sequencing technologies, we've used more or less exclusively FFPE tissues. So our question really was, if we can generate reliable data from these different studies, can we integrate that? Can we integrate fresh frozen Illumina data with Lexogen QuantSeq FFPE data? That was our real question here. So over on the right-hand side here, you can see our Venn diagram, which shows the number of genes after filtering. So our filtering was essentially removing genes which had less than 10 read counts in more than 40% of the data. And what we have here is the overlapping genes across these different technologies, obviously excluding the technologies which are not designed to be whole genome. And we have 7,365 genes in common across these different platforms and data sets. So this is, again, something similar to what I showed you before. We have a Pearson correlation heat map. So if we look at the left-hand triangle first of all, the colors down the left-hand side correspond to the different platforms. So you can see the legends in the middle. Those colors correspond. So the blue is Illumina, and the pink is the QIAGEN QIASeq. And along the top, we have the samples represented as a dendrogram with hierarchical Pearson correlation clustering. And what you can see quite nicely is when we cluster all these samples against each other, and this is really just half of that heat map, we can see clustering within platforms again. So it's much similar to the example I showed near the start. Everything is clustered by its individual platform. What we've done on the right-hand side is applied the combat correction I discussed earlier, and you can see just about down the right-hand side that the colors are all now jumbled up. You can't really determine or decipher particular platforms anymore. So if we zoom right in there and start to look and see exactly what's happening, this is just zooming into that dendrogram. The colors, so the solid band of color under the diagram is, of course, the platforms, and we can't decipher any particular platforms there. But underneath, what's quite interesting is we have the different patients, and these samples are now clustering next to each other, irrespective of whether they are fresh frozen or FFPE. If they're matched, they're clustering near each other. So samples from the same patient are now, after that combat correction, irrespective of their platform or sample tissue type, are now clustering near each other. This is possibly better demonstrated using multidimensional scaling. So essentially, we're using the same genes again, the 7,300 genes. On the left-hand side here, we're doing three-dimensional, multidimensional scaling based on all of those genes. And what we can see is the platforms. They're all clustering by the platforms that they were generated on, as expected, and as we showed in the heat map previously. After the combat correction, we can see that, again, the colors are all jumbled up. Now, it's difficult to interpret that and to know exactly what that means. So we have to start thinking about the biology. And when you color these by whether they are pre-treatment samples, early on treatment samples, or late on treatment samples, we can actually begin to decipher some biology there. You can see the treatment effect with blue in the top left, moving down towards the dark gray, which is the 3-month treatment samples. So we're beginning to see some evidence of true biology. What we can do further is look at those 42 proliferation genes which we previously described as being the most significantly changed in breast cancers which respond well to endocrine therapy. So what we've essentially done here is taken all of the samples from our 11 patients across all the platforms, FFPE and fresh frozen, and we have produced a heat map here, log2 heat map of their expression level of these 42 proliferation genes. And we've ranked that from left to right based on the mean expression level, log2 expression level. So if we start by looking at the colored lines directly above the heat map, these are the different platforms. And you can see that no particular association, there's no particular cluster of one platform or the other. And this is also true of whether the samples were derived from fresh frozen or FFPE material. You can see there's no particular clusters forming there. But if you look at the blue, orange, and black lines near the top, again, based on the expression of these genes, we can see the pre-treatment samples, which we know have the higher proliferation rate, and then we have the early on and late on treatment samples, which cluster towards the right-hand side with lower proliferation expression. And don't forget, proliferation is one of those pathways that we showed changes very early in responding patients. So by 2 weeks, we're beginning to see a reduction in proliferation, which is sustained for the remainder of the treatment, which is clearly demonstrated here, irrespective of what platform we performed the analysis on. So this slide here, one of my last slides, was really because some specific examples, specific comparisons with individual samples. So the first one you can see, we're talking about the integration of fresh frozen tissue. So in this case, these are just plots of all the genes, or of the 7,000 genes represented in all the data sets, with one platform on one axis and a different platform on the other axis. The gray is the association before combat correction, and the pink is afterwards. And you can see the Pearson correlation R values there. So in this first example, we've compared Illumina HT12 with the Affymetrix Clarium S. Again, this was we only did a few samples in this, so it wasn't included in the main analysis. And basically, the take-home message from this slide is that with the help of the combat correction, we're generating strong correlations between matched samples, irrespective of the platform they're performed on. Again, in the lower levels, you can see nanostring versus microarray with some excellent correlations there, and nanostring versus sequencing. As I said before, the nanostring, we only performed a small cohort of 50 genes. So to summarize, essentially, we know that traditional microarrays tend to perform poorly with RNA from FFP tissues, especially older tissues, more than 10 years old. And there are now a range of new approaches and technologies which are available to allow the generation of robust gene expression profiles from formalin-fixed paraffin-embedded tissues without the need to perform costly full RNA sequencing. So we briefly talked about BioSpider, which we haven't used too much, but it's a promising technique. And this is the technique that involves direct sequencing of the RNA. We've talked about nanostring as a potential for smaller groups of genes. It's not available in a whole genome approach, but for smaller groups of genes, it appears to be a suitable way to analyze FFP tissues. In our experience, however, the 3' RNA sequencing approaches, such as the Lexogen QuantSeq and the QIAGEN QIAseq, perform very well for the FFP-derived RNA. Our most extensive experience to date has been with Lexogen QuantSeq, and it's our preferred method to date. We have been able to generate robust, biologically meaningful whole genome transcriptome data from FFP tissues which are over 10 years old. And in fact, the samples that we have performed, we have done so with 95% success rate. Another advantage is that the analysis or the sequencing is included with free analysis on the Bluebee Genomics platform, which makes pre-processing of the data very easy. So I suppose my take-home messages, and as I said before, this wasn't a fully comprehensive study. This is more sharing the experiences of a translational lab with these technologies. We have a range of affordable approaches which are now available for the successful transcriptomic study of FFP tissues. I think, importantly, the selection of technology will depend largely on the question that you're asking and the budget. And another thing that I think we've demonstrated is that data generated from either fresh, frozen, or FFPE tissues using different technologies can be reliably integrated for robust meta-analysis, given the caveats that I've already mentioned. And so if we go back to our original question, can we produce these kinds of data sets from FFPE tissue, and is integration possible? I think the answer is yes, with the caveats that I've mentioned. So I'd just like to thank you all very much for tuning in today, and I hope this has been of some value. Our paper with considerably more data is currently out for review, so hopefully that'll be out shortly. And the data sets have now been uploaded to NCBI Geo if you want to perform your own analysis and look at the data that we've generated. So I'll say thank you very much, and I'm happy to finally just some acknowledgements. Thank you very much to Lexogen for sponsoring this webinar. Thanks very much to the Genomics Core at Edinburgh University, who performed the vast majority of the sequencing that I've shown you today. Some of our collaborators in North Carolina and Georgetown, elsewhere here in the university, my own lab team, of course, bioinformatics input from Dr. Andrew Sims and Dr. Chidam Selly, and of course our clinical team. And finally, our patients who make all of our research possible. So thank you very much, and I will happily take any questions.
Great. Thank you, Dr. Turnbull. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right. Arran, our first question, would the correction methods that you talked about work if there's a combination of different subtypes in the samples that are being analyzed, for example, ER2B mixed with ER negative?
That's a very important question, and this, I suppose, I didn't go into this in too much detail. This really goes back to the point I mentioned about selecting appropriate data sets to combine. What we've shown before is that if you have all of one particular biological feature in one data set and a different subset enriched in a second data set, the correction methods sometimes are not appropriate in that case. So really, we've aimed to always integrate studies which are similar in their composition. Obviously, that's a very difficult thing to think about. In terms of the breast cancer intrinsic subtypes, we performed the analysis independently in those two public data sets that I showed first of all, and determined that their composition was such that they could be reliably integrated. I think if we'd found an enrichment of one particular group, it may not have been appropriate to combine those. So again, it's a very complex question, but it's something that requires a lot of thought.
So you mentioned several correction methods in your presentation. Do you have a specific one that you prefer in order to reduce systemic bias in FFPE samples?
So I personally, I've used a lot of these for a long time now. My two preferences would be XPN, or cross-platform normalization, and combat, both excellent approaches. One of the advantages of combat is that, as I said before, you can have unbalanced data sets. For example, you can have a data set with a smaller sample size and a larger sample size and still effectively reduce the bias. You can also combine, so for example, in this data set, we were actually combining several data sets from different studies, and in reality you probably wouldn't do that. This has been more of a technical thing that we've done here when we were integrating 7 different platforms or something, more to show that it's possible as an example. So the one advantage is that we did that all with one iteration of combat, which would not be possible with XPN. So yeah, we have extensive familiarity with both, and we've used both quite extensively. Those two would be my preference.
Can you talk a bit about detecting alternative splicing events in FFPE samples?
Actually, probably not. That's something we haven't yet looked at. It's something we are interested in going forward to do, but it's not something that we have looked at yet. So our full RNA sequencing data that we generated is relatively new, and we haven't really got to grips with the analysis of that yet. So it's not something we've done, but it's something we're going on to do next.
And apart from the technical quality control recommended by the manufacturer, can you give us a bit of, can you talk a little bit about what the evidence was that whole transcriptome analysis was meaningful for 10-year-old FFPE samples?
So yeah, essentially, what we basically did is we've basically taken analysis of high-quality, fresh, frozen RNA-derived microarray data that we've produced, over the last few years, that we have extensively analyzed. And what we basically did is applied that analysis to these FFPE tissues. So, the example I gave here, of course, was just proliferation. We've actually extensively studied the effects that we see with treatment, not only in patients who respond, but in patients who do not respond, where we see completely different profiles. And we've actually been able to replicate all those analyses in this cohort of FFPE samples, in our larger cohort of Lexogen FFPE samples. One of the other things that we have done is analyzed some gene expression data sets which we know relate to particular clinical outcomes, and demonstrated that with some of the follow-up data that we have for these older FFPE samples.
In your experience, do the different platforms that you talked about show differences in terms of reproducibility of data, and are different platforms more susceptible to batch effects?
Again, that's a very good question. I think, to be honest, we haven't processed enough samples on enough platforms to make, to be able to infer that. We've basically approached this from the point of view, thinking about where systematic bias can stem from, and shown that different technologies are obviously the biggest contributing factors to systematic bias. What we have not looked at is the contribution of specific platforms to that, simply because we don't really have the data. As I said, this is not a comprehensive analysis. It wasn't intended to be that. We don't have enough samples, enough of the same samples on the same platforms to do that kind of analysis. And for some of the technologies, such as BioSpider, Seek, at the moment we have only processed a very small cohort of patients on those platforms. So we're not really able to draw too many conclusions yet.
Do you have any recommendations for subtyping a single breast cancer sample, since batch effect correction probably wouldn't work with that?
Again, that's a very good question. We have used a number of R packages, such as the GeneFu R package, which allows single sample assignment to subtypes. The answer to your question is I'm not entirely sure if we yet have an established reliable method for that. We know that if we use different sets of genes and we have different compositions of the subtypes within a particular data set, that we can assign samples to different subgroups. We know that different bioinformaticians can assign samples to different subgroups. So the idea of subtyping is very, very complex, and that's why, for example, for tests that rely on subtyping, such as the Prosigna test, this is performed on one particular technology, which is the nanostring technology, in order to increase reproducibility. We have showed, not data here, but we have showed that using the PAM50 genes, which are the input for the Prosigna data generated in different technologies, we can actually have different assignment to subtypes. So it's a very complex question, and I don't think we have a reliable method yet that can be used across the board and reproducible across different data sets.
Were you able to derive RNA integrity numbers, or DV200 values, for the FFPE samples? And if so, what was the quality of your RNA samples from FFPE?
That's another great question. So yes, we routinely do that. So, of course, going back to our fresh frozen tissue, We had usually high RIN numbers of 7, 8. We routinely see, especially in older FFPE samples, and we did an analysis a while back looking at the effect of age on FFPE, we routinely see RNA values as low as 2 and 1. We were, when we were performing the AmpliSeq approach, we were using the 200 nucleotide degradation QC as an input for that. So we were looking for things obviously above 200. We even had some fails above the 200 mark. So we decided that wasn't really the best technology for some of these more degraded samples. We have since looked at that same metric in our other samples, and irrespective of what the results were, we decided to go ahead and process them with the Lexogen platform, and were able to derive robust, usable data, which again, it was the input to our comprehensive breast cancer biology analysis. We were simply checking to see if the biology we knew should be there could be elucidated from those samples, and it could. Proliferation being one example that I gave in this talk.
Did you use any modified protocols for FFPE samples on the QuantSeq, and did you count intronic reads as part of the expression analysis?
No, we performed the analysis exactly via the manufacturer's protocol. Again, for specific comments on that, I'll refer you to the Genome Core, but it was performed as per the standard protocol and as per the standard analysis pipeline through the Bluebee platform.
It seems like the goal has long been to get good quality data from FFPE samples, and you talked about some of the ways that can be done. But is there a better way to preserve tissue, or is freezing the only alternative?
Again, that's a very interesting question. I think, to be honest, we, especially in a clinical environment, I think it's quite difficult to logistically introduce something that's not done as standard. The advantage of FFPE is, of course, that these tissues are collected and processed through routine pathology labs and are therefore stored as part of the normal healthcare process. We are quite lucky here in that we have support staff that do collect fresh frozen tissue for us. But the logistics and challenges involved in that include things like taking liquid nitrogen into theaters in order to collect the tissue and preserve that immediately, because obviously RNAs begin to degrade very, very quickly. And so it is a logistical challenge. We have never really looked at anything else other than comparing high-quality fresh frozen tissue, in which, in the case where we were essentially, as I said, we took liquid nitrogen into theater or the clinical room and collected the biopsies directly as our sort of gold standard, and then FFP on the other side. We've never looked at anything else.
Since you used only the overlapping set of transcripts to correct for bias between data sets, can you still analyze the transcripts that are not included in this common set?
Sorry, you just cut out a little bit there. Could you repeat that?
Sure. Since you used only the overlapping set of transcripts to correct for bias between data sets, can you still analyze the transcripts that are not included in the common set?
Yes, and in fact, we actually did that. As I said before, that we weren't planning on doing the purpose of this was not a sort of a comprehensive study. We were actually generating data to address specific research aims of our own. So we analyzed all of these data sets. Or a number of these data sets, specifically the large Lexogen data sets, absolutely independently, and then included all the transcripts, not just those that were common to all the other platforms. And most of the analysis that we have done, and essentially replicating the analysis that we performed previously, or the characterization of endocrine therapy response we performed previously on fresh frozen tissue, we were able to replicate that using the whole genome QuantSeq data.
Did you use the same RNA extraction kit for all the platforms? Was it whole section or macro dissection? And can you tell us if there was a cutoff in the percentage of tumor in the samples?
Again, that's an excellent question. So in this set here, what we actually, we used two kits. We used the AllPrep QIAGEN FFPE kit, and we used the mRNA-Seq QIAGEN kit for the fresh frozen tissue. So all of the samples were processed via one of those two kits. In terms of our sample selection, we do not routinely perform microdissection. So we start by doing an H&E analysis of the tissue, and we exclude cases which don't have sufficient tissue, so or don't have sufficient tumor content, I should say. So in this context here, we had 60 to 80% was our lowest. So anything higher than 60% tumor content went forward here. So all of our samples had that approach. For some of our biopsy tissues, the minimum was a single biopsy. So we're using relatively small amounts of tissue here in some of these cases. Obviously, with fresh frozen, we have a lot more.
What was the sequencing depth by QuantSeq for FFPE samples?
I don't have that data to hand. I would need to check that, so I can get back to you on that. I don't have the QC metrics with me here.
Okay, great. And what are the ranges of necessary RNA input for your preferred RNA-Seq platforms?
So again, this varies from platform to platform, and probably the genomics core is the best people to speak to with this. What we have done, some experiments before using universal human reference RNA, which has been diluted down, and we've put in order to generate, in order to see if the actual input amount can affect the results, and we have pretty high correlations in that set. What we have done here, mainly because of the available tissue that we have, is that we put in the highest recommended RNA quantity into these studies. So for the, I remember off the top of my head, for the Lexogen QuantSeq study, we put in 500 nanograms total of RNA extracted from the FFPE tissues.
Do you have any experience collecting samples with RNA Later sample collection kits? And if so, could you tell us a bit about it?
So we have used RNA Later in the past. We've also used RNA Later ICE in the past, and it was working quite nicely initially. And I'm actually going back about 9-10 years ago when we last used this. But we haven't really used either of those approaches since then. When we first embarked down the FFPE route, we looked through a range of different FFPE extraction kits and approaches, and did a whole set of QC with Agilent and Qubit, etc., and determined that the QIAGEN kits were highly reproducible and gave us good quantity and quality of RNA compared to some of the other kits. In terms of the fresh frozen tissue, we have always gone for we essentially freeze in liquid nitrogen, pulverize the tissue, and then put that straight into the mRNA-Seq QIASeq kit. So that becomes the input to that. We've always processed the fresh frozen tissues in that way, and we've always had high quality, abundant RNA.
Are there any test data sets available in the public domain that can be analyzed?
Yes, another great question. What our paper is actually kind of out for review, which has a little bit of what I've talked about today and some more analysis. And all of the data sets are now currently on NCBI Geo, so they should be available shortly. If not, they're definitely in that paper. So they should be available to be used.
Great. Thank you, Dr. Turnbull. We've reached the hour, so we're going to wrap it there for today. We'd like to thank Arran Turnbull and our sponsor, Lexogen. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. And if you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.